So in chapter 7, we talked about a lot of the external requirements that organisms need to thrive, whether it be different elements, energy sources, oxygen requirements, relationships with each others. It's what are those external things that they need to survive. Chapter 8, we're going to go inside the cell. So once those organisms have everything they need, what's happening inside those cells, what's happening to some of the nutrients that gets in there, what's happening inside the cell. So I'm going to first kind of just I'm going to make it look a little bigger so we can take a look at, I like the scope, that first part of the PowerPoint is what are the main topics that this chapter is going to go over. It's the enzymes that are needed for the chemical reactions that are happening in there. What are some of the metabolic pathways that happen for different reactions? What are some of the sources of energy? And then we're going to talk about anabolic reactions and pathways and catabolic reactions and pathways. And then, the chemical reactions that allow us to break down bonds as well as build things up. And it's going to be talking about how we get energy, how do we get ATP from breaking things down, and specifically how do we break down sugar, like glucose, to get us a lot of ATP. So we're going to talk about glycolysis and the Krebs cycle and electron transport chain. Now, if you've already had taken advanced A&P, this will be a nice review because you go over those same type of steps. If you haven't taken A&P yet, advanced A&P, this will be a preview. And if you're taking it at this exact same time, sometimes the units kind of go at the exact same time as each other. So you might be hearing about Krebs cycle and electron transport chain from both ways. My only note, though, is when you talk about glycolysis and Krebs cycle and electron transport chain in anatomy and physiology, that's all happening in a eukaryotic cell, because that's all we have. Prokaryotic cell does have some differences, though, in the steps. And I don't go into as far as depth of the Krebs cycle and electron transport chain as Anat and Phys does. But we're going to start off in this chapter is talking about some terms when we're talking about different chemical reactions happening in the cell. So the word metabolism, which you may have heard just out and about when people talk about, oh, I have a fast metabolism or a slow metabolism. The word metabolism just means all of the chemical and physical processes happening in the cell. Everything that's getting broken down, everything that's getting built up. So if someone says they have a fast metabolism, it means all of those reactions are happening quickly and sometimes more efficiently. Now, we do break down all of those chemical reactions because metabolism is all the processes. Catabolism means when we're breaking things down. So we're breaking bonds, we're taking large molecules, we now form smaller molecules. The opposite, anabolism, is where we're going to make larger things. We're going to take smaller molecules and we're going to form larger molecules. That requires energy. If you're going to build a building, you need energy. You need the building blocks. You need things to put it all together, and you need energy to build it. Breaking things down does not take as much energy as building things up. Now, you got to know the difference between a catabolic reaction and an anabolic reaction. And my way to help try to remember them is if you know anyone that's have a cat, maybe you have a cat, but cats like to break things. They knock things over all the time. So I always just think catabolic reaction is you're breaking things down. So you're breaking things. And then you just have to remember that anabolic reaction means the opposite. So cats break things. So catabolic reaction is breaking things down. Now, when things get inside of our cell, we've got a little ciliated cell here. Things get inside of it. When you break things down, it releases energy. So catabolism, breaking things down, it releases energy in the form of ATP. But an anabolic reaction can then use that energy to build things up. So it's constantly going back and forth. We're breaking things down to constantly build things up. One thing that helps in all of these chemical reactions, because there are millions of chemical reactions happening in a cell at any particular time, lots of things getting broken down, lots of things getting built up. One thing that helps all of those reactions to happen are enzymes. Now, an enzyme, it's a biological catalyst that increases the rate of a reaction by lowering the energy of activation, which is really wordy, but I'm going to tell you what that means. So it is going to help speed up chemical reactions and help make those chemical reactions happening. Now, a note about enzymes. Enzymes themselves don't get altered in the reaction. So the enzymes don't change. Instead, they just promote some process for happening. Now, an example, how enzymes work. If we have some type of reactant and we want to make a product out of it, and in this one, I'm going to break this big thing down into the two individual things. Before I can get any reaction to happen, because they don't spontaneously happen, before I can get any reaction to happen, I first have to put some energy in. Before I can get this rock to go down the hill, I first have to put some energy in to get the rock moving. Now, if it takes a lot of energy to make that happen, sometimes reactions don't happen. So what enzymes do, so this is the amount of energy on this diagram that is needed. So here's my initial. This is the amount of energy that's needed if there wasn't an enzyme. If an enzyme is present, it lowers the amount of activation energy that's needed, which means it doesn't take as much energy. It might just take a little tiny shove and that rock goes over the hill. Now, I found this from a different site, so it's not in your textbook picture, but I liked it because I like jumping frogs. So if these are my reactant frogs and they want to become product frogs, this is the activation energy barrier. This is how much energy they need to jump over to become a product, which they might not have enough energy to make that happen. However, if there's an enzyme, it lowers that barrier. It lowers how much energy is needed. It's a lot easier for those frogs to jump over, and so it is more likely that a reaction will happen. So when we go back, they are catalysts. That means they're going to speed up a reaction. They increase the rate that a reaction is going to happen by lowering that energy of activation. They lower that barrier so that reactions are more likely to happen. Now, enzymes, found these also from different sites, enzymes do have an active site. It's where the substrate that they're going to work with or work on bind. Now, on this particular one, because generally most enzymes, which they are proteins, everything seems to be proteins, enzymes and proteins seem to be like big blue and big purple blobs in any textbook. And this is no exception. Any type of enzyme has an active site. That is where the substrate is going to fit. And it's going to be specific. Nothing except this particular molecule is going to fit in there. Now, my note, everyone's like, but it doesn't fit completely. This is round and this is square. True, but if they are specific for each other, that this will fit in there. And once a substrate gets into the active site, it does form a more perfect fit. And that enzyme is going to work on that substrate. Now, this is showing an example of another enzyme that's going to work on this substrate. It's a type of sugar. It's a fructose sugar. As soon as it gets into that enzyme, in the active site, it makes a more perfect fit. That enzyme breaks apart that sugar, and then the enzyme's right back there again. So the enzyme doesn't get changed. The enzyme doesn't get used up. It just gets recycled. It can be used over and over and over again, doing whatever it is that's supposed to do. This particular enzyme is specific for this particular sugar to break it down. Now, a lot of times enzymes are named for the substrate that they work with. So this substrate, this sugar, fructose 1,6-biphosphate, fits into the enzyme. The enzyme's called fructose 1,6-biphosphate aldolase. Most enzymes, about 99% of them, all end in ase. So if you ever read anything and it ends in an ase, it's a pretty good clue it's some type of enzyme. They aren't always named for their substrate, but this is a good example of one. Now, an enzyme is a protein, and so things can happen to enzymes to make them not quite as efficient. So if the temperature changes, pH, osmotic pressure, and like that can all change an enzyme, which is a protein. And anytime you change a protein, it's called denaturation. It's not going to work correctly. And so if we change the pH, if we change the temperature, enzymes aren't going to work correctly. And if those enzymes don't work correctly, you might not get chemical reactions to happen because we need those enzymes to lower activation energy. Now, with all of these different chemical reactions that happen, there are a lot of metabolic pathways. It just means that you've got different reactions that happen in a sequence, that one thing becomes another thing, and that product that was just made goes into another reaction to become something else. And the product from that reaction goes into another reaction and becomes something else. And so we've got a lot of pathways to eventually give us our end product. And they usually form very specific pathways to maximize the use of any available nutrients and energy. And I'm like, and we keep all of it in check. Again, our cells try to be, not just our cells, bacteria cells too, try to be as efficient as possible with whatever is available. Now, enzymes, there can be some things that can mess up enzymes, not just temperature and pH. There are other things out there that can change enzymes. And anytime you change an enzyme, it's never for good. And they're called inhibitors because they're going to inhibit the enzyme, which is going to inhibit chemical reactions from taking place. Now, competitive inhibition means you're going to have some substance that resembles the substrate that's supposed to fit in the active site, and so it competes for the active site. Now, this is showing a picture that here's your enzyme, big blue blob. This is the substrate that's supposed to fit in there, but the inhibitor resembles the substrate. It's not an exact copy, but it resembles the substrate, and it resembles it enough that it can fit into the active site. And if this substance, this inhibitor is in that active site, the substrate can't fit in there and nothing's going to happen to the substrate. An allosteric inhibitor means that you're going to have some substance, some type of inhibitor that's going to bind to the enzyme, but in a different spot other than the active site. Some enzymes have two binding spots. They have their active site binding spot, and they have another spot called the allosteric site. Picture. So if here's your enzyme, this is the substrate that's supposed to fit in there, but here's an allosteric site. If that inhibitor fits into that other spot, it can make this enzyme change shape. And the change shape now is that at the little top here kind of flips over. And that little bit of change means the substrate can't fit in there anymore. And if the substrate can't fit, can't break it down or whatever it's supposed to do with it, and again, it's an inhibitor. It has stopped the enzyme. It has stopped the chemical reaction from happening. Now, feedback inhibition. So this is showing like a biochemical pathway that one substrate goes into an enzyme, becomes a product. That product goes into another enzyme, becomes a different product, and so on. But occasionally, your final end product, whatever it is that's supposed to be made inside the cell, that end product can sometimes be an inhibitor. That little green oval can go back up to that first enzyme, fit into another spot, an allosteric spot, the enzyme changes, and that initial substrate can't fit in there anymore. This happens quite often when cells, their job is to make an end product, whatever it is supposed to be, and once they've made it, they don't want to make any more. They want to make just enough that they need, and they don't want to make any more. So a lot of times that end product becomes an inhibitor so that just the right amount is made. Now, energy. The term itself is just the capacity to do work or cause something to change. And there's different forms of energy. For us, we're going to be looking at when we break things down, we release energy. When we build things up, we require energy. And energy is used for lots of different things. Now, an endergonic reaction just means some type of reaction that requires energy. Energy goes in. Endo or ender means goes into. So energy has to go into a reaction. This is usually when you are building something up. An exergonic reaction means you're going to release energy. Anything ex or exo means outside, so you're releasing energy. An endergonic reaction that requires energy is usually some type of anabolic reaction. You're building things up. An exergonic reaction where you release energy is usually some type of catabolic reaction. Now, when we're constantly breaking things and constantly building things inside of cells, when we break things to release energy, energy itself is usually tied to an electron. Electrons, those negatively charged particles, carry energy. So they themselves have energy. So a lot of times when we're talking about energy, we got to talk about electrons as well. Now, when you have the breaking of a bond and you release energy, you release an electron. Electrons don't like to just hang out, floating around anywhere. They're unstable. So they usually, right away, as they get released, go somewhere else and bind to something else. So when we follow the electrons that are getting released and then going somewhere else and binding somewhere else, we call it a redox reaction, a reduction oxidation reaction. It's just short for reduction and oxidation. We smush the words together and we have a redox. So it occurs in pairs, where when you release an electron, there's something right there to gain an electron. So on this picture, I have this in our reaction, A is losing an electron. And when you lose an electron, it's called oxidation. And when you lose an electron, because an electron has a negative charge to it, the result, you'll have a positively charged molecule at the end because it lost an electron. But you also have another substance there that's right there to gain that electron. Because when you lose an electron, something right there, my B, is there to gain an electron. And after it's gained an electron, which is called reduction, it is now negatively charged. Now, this is where it usually confuses students. When you gain an electron, it's called reduction, which sounds like an oxymoron, because when you become reduced, you're usually not gaining something. However, in this instance, you got to know what you're gaining. You're gaining something negative. And so when you gain something negative, you become reduced. My analogy for it is if you gain a bunch of negative friends, you become reduced or more negative yourself. So that's why reduction means gaining, because you're gaining something negatively charged. Now again, we care about electrons. They carry energy. So electrons are highly reactive, and they are usually captured and transported around. They have energy in them. We need to get that energy to somewhere else. And so they're usually attached with usually a hydrogen atom, and they can get shuttled around, and there are 3 top electron carriers. So these are molecules that readily pick up those electrons that are released, and they usually pick up a hydrogen atom with it. And the top 3 electron carriers, NAD+, when it picks up an electron, it loses that positive charge, but it also picks up a hydrogen. So NADH means that it is an electron carrier. I do not care that you know what NAD stands for, the nicotinamide adenine dinucleotide, as long as you are familiar with the abbreviations as being electron carriers. So NADH, NADPH, and FADH2 are our top 3 electron carriers. They are going to be right there waiting for an electron, and I'm like to shuttle somewhere else. Now, electron carriers, this is just showing that NAD+ picks up an electron, a couple electrons, as well as hydrogen on the molecules. Now, I did put in the little concept check. I left one on here. If a molecule has been reduced during a reaction, because this is that tricky part. If it's reduced, it has gained electrons, and it usually has also gained those hydrogens as well. Now, usually when we talk about energy, it's not just talking about electrons. We usually talk about ATP. It is the energy currency of a cell. It's made up of adenine, ribose, and a 3-phosphate group, hence the triphosphate part. Now, that last phosphate is constantly removed. There is energy in the bond that holds that last phosphate, holds any of the phosphates on. And when we remove that energy, when we break that bond of that last phosphate, it releases energy. So ATP has stored energy in the bonds. Here's my picture. and like your adenine, your ribose, and your 3 phosphate groups. Again, the bonds that hold the phosphates together have lots of energy. You release or break that bond, you release the energy. And if you release that last phosphate, it then becomes adenosine diphosphate. You can also release a second phosphate, then it's adenosine monophosphate. Usually just one phosphate is released. And so removing that last phosphate releases lots of energy. We'll go forward. And so there are lots of processes that help make ATP by adding an inorganic phosphate onto ADP, your diphosphate. And so when you put that last phosphate on, that third phosphate onto ATP and get ATP, we can use the energy that's stored in there now to do some other type of work in the cell. Now, adding that last phosphate on is called phosphorylation. It's taking energy that was made somewhere else in the cell, whether it was breaking something else down. Now, maybe depending on the cell itself, it could have been because of some type of photosynthesis or light energy, but it's taking that last phosphate group and adding it to ADP. And it's just phosphorylation. Now, taking all of this that we've just gone over, we're going to get to now kind of the heart of the chapter, as we've got a lot of catabolic, breaking things down, and anabolic reactions, breaking things down and building things up. One of the biggest ways where we can get lots of ATP, so we can do lots of work with it, we can get lots of ATP, and we're going to get a lot of electron carriers that help make a lot of ATP from the breakdown of sugar, goes through a series of 3 main steps: the glycolysis step, the Krebs cycle, and the electron transport chain at the end. Now, again, this is where we're going to get some differences between a eukaryotic cell and a prokaryotic cell, is because prokaryotic cells are a little simpler. They're going to do the basics of this, but with a few changes. Now, we're going to kind of break down the steps. They're going to be similar steps, but we're going to break down some of the steps and talk about how cells can do this aerobically, anaerobically, and how they can do part of the steps through a process called fermentation. All of it is going to start off with sugar or glucose at the very beginning, and all of the steps are going to give us ATP, going to give us some type of energy. So in your book, they've got a nice little breakdown of each: aerobic, anaerobic. They almost look the same, and there are a lot of similarities. And then fermentation, which doesn't take it quite so far. But either way, they all start off with glycolysis, with glucose, and we're going to get carbon dioxide, and we're going to get energy. We're going to get ATP out of any of these 3 steps. So we're going to first start talking about going through the process aerobically. So oxygen is present. The cells are going to utilize the oxygen that's present. Now it's going to start off with glycolysis, which is going to get split. The word, or that suffix lysis, means split. Glyc means sugar, so glycolysis literally means splitting sugar. We're going to split glucose. Now, I was going to say, here's the process of glycolysis. You do not have to know this whole process of glycolysis. Please, you do not have to know this whole process of glycolysis. If this stuff interests you, I know our local university, UW La Crosse, they offer a whole class just on cells. It's called cell biology. You can spend like 3 weeks on glycolysis and have at it. I mostly care that you know the basics. What goes in, what comes out. What do we get out of this process? So for what goes in, because we're just looking at this first step through aerobic respiration, is the glycolysis. Glucose is going to go in. Pyruvate or pyruvic acid is going to come out. Different books called it different things. Pyruvate, pyruvic acid, same thing. And you're going to take a 6-carbon molecule, because glucose is C6, H12O6, and we're going to break it down into 2 3-carbon molecules called pyruvic acid or pyruvate. What I care about is that we get some ATP and we get NADH. Why do I care about NADH? It's an electron carrier. It means it's carrying electrons. Now, what what we get out of it for ATP. At the very, very top of this process, 2 ATP have to go in. Reason why? You need some activation energy. Reactions don't just happen spontaneously. You need a little energy to get these things started off. So we're going to put a couple ATP in. Then we're going to take that 6-carbon sugar. We're going to split it. Going to do lots of things to it. Don't care about. But in the end, I'm going to get 2 3-carbon molecules called pyruvic acid. I'm also going to get some ATP out of here. I get 2 ATP here, and I get another 2 ATP here. So I've made 4 ATP through glycolysis. Cost me 2 ATP, but I made 4. So we say that you net 2 ATP. But the couple other things we get out of here is we get some NADHs as well. We get some electron carriers out of the process. Now, your 2 pyruvic acid, your 2 3-carbon molecules then go into the next step. So it's called the fate of the pyruvate. Was we're going to take that pyruvic acid, pyruvate, same thing, and it's going to get further broken down. So we just did glycolysis, we just got pyruvic acid, there's your 3 carbons, and it's going to get further broken down into something called acetyl-CoA. Now, out of taking pyruvate and making acetyl-CoA, stands for acetyl coenzyme A, we do get a little carbon dioxide out, but we get another NADH here for each pyruvate. So we get a couple more NADHs, more electron carriers, more electrons. And then that acetyl-CoA can now go into the Krebs cycle. Now again, your book, I think I deleted your Krebs cycle book because I didn't like it. And I'm like, this seemed a little simpler when I made it larger. Again, lots of things happening in the Krebs cycle, a lot of things I don't really care that you need to know. I care again about the basic, what goes in and what do we get out of it. So acetyl-CoA goes in, and you've got 2 acetyl-CoAs because you had 2 pyruvic acids, you've got 2 acetyl-CoAs, so you really have 2 Krebs cycles that are going to happen. Acetyl-CoA, it's a 2-carbon molecule. It's going to find this 4-carbon molecule called oxaloacetic acid or oxaloacetate. You put them together and they make a 6-carbon molecule called citric acid. I was like, oh, you're right back to a 6-carbon molecule. You are. Structurally, it's not glucose, but you are back to a 6-carbon molecule. Now, it's called citric acid, which is also why the Krebs cycle is sometimes called the citric acid cycle. Now, this 6-carbon molecule, lots of reactions are happening to it until it eventually goes back to your 4-carbon molecule. So you lose a couple carbons. Where'd they go? Well, one of them's lost over here as CO2, and there's the other one that's lost to CO2. But the interesting part about the Krebs cycle is the other stuff we get out. We get out NADH. How many? Well, we get one NADH here, we get another NADH here, and we get another one way up here. We get 3 NADH out of each acetyl-CoA, so a total of 6, because you have 2 acetyl-CoAs. You're also going to get out FADH2, another electron carrier. Just 1 out of each Krebs cycle, so a total of 2. And we get some ATP. Just 1. But you've got 2 Krebs cycles that are going to happen, so we get 2 ATP. So we get some energy, and we get a whole ton of electron carriers out of the Krebs cycle. Now again, you've got 6 NADHs, 3 from this Krebs cycle and 3 of them from another Krebs cycle. So 6 NADHs, 2 FADH2s, and more ATP. Lots of energy and electron carriers that are happening with all of the processes in the Krebs cycle. Now, I found this somewhere, and I'm like, ignore where it says see worksheet. Sorry, I forgot to delete that. And I'm like, where the Krebs cycle takes place. The Krebs cycle, depending on the cell, takes place in the cytoplasm. And I'm like, we haven't gotten to the, it happens, I guess, whether prokaryotic cell or eukaryotic cell, it's going to happen in the cytoplasm. Now, the what goes in? Well, acetyl-CoA goes in. 2 of them. And I'm like, what comes out? We get a lot of NADH. We get a lot of FADH2. Well, 2 FADH2. We get a lot of electron carriers, and we get some ATP out. And that's kind of my, what's the anything interesting, important part is that we get a lot of NADH and we get a lot of FADH2. And a lot of these in-between molecules that I don't care that you know the names of, usually are a lot of precursor molecules. They're building blocks that can get used elsewhere for other processes. Now, once we've got all those electron carriers, we've got ATP, we've got some precursor molecules, we get to the last step, the electron transport chain and oxidative phosphorylation, is we're going to finally know what's going to happen to all those electron carriers. So it's a chain of reaction that is going to take that energy in those electrons and the hydrogen to churn out a whole ton of ATP. Now, in your book, where this happens, well, this happens in the mitochondria. However, prokaryotic cells, way too simple. They don't have a mitochondria. So this has to happen out on the cell membrane. So if you notice, this happens on the inner matrix. The majority of this happens on the inner matrix of the mitochondria, phospholipid bilayer. Well, bacteria cells, they have a cell membrane, phospholipid bilayer, and so this is going to happen out on the cell membrane. Now, I'm going to say the book gets kind of wordy on here. I like to go through diagrams. You can read through it. You can flip back and forth. I want the picture because I found this picture that seemed a little simpler and was zoomed in a little bit more. So whether this is happening on the inner membrane of the mitochondria or whether this is happening on an outer membrane, it's phospholipid bilayer. Now, all those electron carriers that we created in glycolysis, whether we created them making acetyl-CoA or whether they came from the Krebs cycle, are all going to head to the electron transport chain. Now, the energy, the electrons that are in those electron carriers are going to be used, that energy is going to be used to pump the hydrogen ions across the membrane. But then all of a sudden we end up with a whole bunch of hydrogen ions on one side. And just like you guys, no one likes to be crowded, so they're going to try to go back to the other side. But what we've created here with the energy in those electrons, we've created a hydrogen gradient, a hydrogen ion gradient, where you have lots of hydrogens on one side and not a lot of hydrogens on the other side. So what those hydrogens can do when they want to sneak back to the other side is they're going to find an enzyme, enzynase. It's called ATP synthase. It's going to tell you what it does. And they can sneak back through that enzyme to go to the other side. However, when they do that, they turn, this top part is kind of like a turbine. They turn that enzyme. And just like a wind turbine or a water turbine, when you spin it, it generates energy. Well, when that hydrogen ion sneaks through and changes the enzyme, when it spins around, it generates energy for phosphorylation, to add that phosphate onto ADP, and it now makes ATP. So you have lots of electrons that are pumping lots of hydrogens across, and lots of hydrogen ions are trying to sneak back, and that enzyme ATP synthase makes lots of ATP. Now, this again is just showing that here we've got electrons, again, it's all written out. They're pumping the hydrogen ions across. You've got that gradient, that difference of hydrogen ions. They again want to go back through, cause it to rotate, make lots of ATP. Now again, bacteria that don't have a mitochondria, it's going to have to happen out on the cell membrane. Again, same thing, energy in the electrons is going to pump hydrogens across, and the hydrogens are going to try to sneak back through that ATP synthase enzyme. Now, but the electrons, they do have energy, but they still have to end up somewhere. So we use the energy to pump hydrogen ions across in this last step, but then where do they go? What's the final step? Well, in aerobic respiration, where oxygen is plentiful, oxygen is the final electron acceptor. So that final electron plus some hydrogen ions that are around are going to go find an oxygen, and when they do the balanced chemical equation, they end up with water. An aerobic metabolism, this whole process of the glycolysis, Krebs cycle, acetyl-CoA, electron transport chain, does give us a maximum yield of ATP. on average, cells can make 38 ATP for just one molecule of glucose. Now, that can change though. Different organisms are more efficient than other organisms. And part of that is some organisms have leaky membranes. And if that hydrogen ion sneaks back across without going through the ATP synthase, you're going to have a lower yield of ATP in the end. Now, this again is just ATP. In theory, you get about 38 ATP. However, anaerobic respiration. I like the Big Bang Theory. You may find a few more memes throughout my PowerPoints. Anaerobic respiration, again, there's no oxygen present. And these organisms, they don't require oxygen. Sometimes oxygen's toxic for them, but they can still do a lot of this. And I'm like, it generally isn't quite as efficient. You usually aren't going to get that maximum number of ATP out. So they're still going to do aerobic respiration. They're still going to do glycolysis. But some bacteria that are anaerobic, you guys are not anaerobic, but some bacteria that are anaerobic can still do glycolysis, acetyl-CoA, Krebs cycle, and electron transport chain anaerobically. You can't, but some bacteria can that are anaerobic organisms. My note on anaerobic organisms, when you get to the end, again, you can still do glycolysis, acetyl-CoA, Krebs cycle, electron transport chain. The only big difference is that final electron acceptor. For aerobic respiration, the final electron acceptor was oxygen, but not an anaerobic respiration. So they have to find some non-oxygen electron acceptor. Now, everyone's like, but those are oxygen. Yes. And I'm like, these are technically, but although they have oxygen in them, they are not a straight up standalone oxygen molecule. And I'm like, they may have it in their formula. If you're in an environment and the only thing in the air is carbon dioxide, CO2, you're in an anaerobic environment because there's no free oxygen. So even though oxygen's in the chemical formula, it's not all by itself. And the last way of breaking down sugar is fermentation. Now, the final electron acceptor is going to use an organic compound, so some type of carbon-hydrogen molecule. It yields the smallest amount of ATP because it doesn't go much farther than glycolysis. Glycolysis gives you 2 ATP, and a net of 2 ATP. You make 4, but you use 2. But you still at least get some energy out of it. And I'm like, it's still breaking down sugar. You're still getting ATP. So it's still talked about. It's in the same process. But the final product is going to be different because the sugar is not going to be completely broken down and released as CO2. You get a product. You get an organic product. Now, depending on the organism, usually depends on what the final product is going to be from that sugar. Some organisms can make alcohol. Some organisms make various types of acids, but you usually do get some type of CO2. So again, it's still going to take glycolysis. It's still going to do fermentation. You yield you a net 2 ATP. And you're still usually going to get some NADH and some carbon dioxide. But then that pyruvate, it just depends on the organism, depends on what kind of final product is made. And I'm like, this again is showing, here's glucose, here's your pyruvic acid. Some organisms can make ethyl alcohol. Some can make lactic acid. Which ones do you make when you work out? Well, you guys make lactic acid when you work out. If your cells have gone into an anaerobic environment, they're functioning anaerobically for a short period of time, they can do fermentation. The main reason you know, or the main way you know that you've made a lactic acid is you're really sore the next day. However, some organisms don't make lactic acid. They make ethyl alcohol when they go through fermentation. That's your basic alcohol. Now, it would be interesting, quite interesting, is if our cells did make alcohol when they worked out. Going to the gym might be a little more entertaining if you made ethyl alcohol instead of lactic acid. But this is to show you some different examples of different organisms and what they can make with that pyruvic acid. So Propionibacterium takes that pyruvic acid and it makes carbon dioxide as well as another type of acid, the propionic acid. That's where we get Swiss cheese. There is an organism in there. The bubbles, the holes in your Swiss cheese is because of all the carbon dioxide bubbles that are released through fermentation. And that acid gives it that sharp flavor. Aspergillus, lactobacillus, and streptococcus can all make lactic acid like us, and it's what gives flavoring. It gives the sharp flavor to cheddar cheese, to yogurt, and to soy sauce. The Saccharomyces, and this is where we get all of our ethyl alcohol. Clostridium, the same genus as C. diff, same different bacteria though, can use pyruvic acid, and their final product is acetone or isopropyl alcohol. So different organisms can make different products at the end of fermentation. Now, in this table is just showing what are the different pathways that it can go through. Again, aerobic respiration can go through all three. Same thing with anaerobic respiration. But fermentation, and I'm like, just does glycolysis. The final electron acceptor is an organic molecule. Final electron acceptor is oxygen, free oxygen. Anaerobic respiration, as I say, I feel like this got mixed up a little bit. Anaerobic respiration finds some final electron acceptor that's not free oxygen. Again, we get ATP out of all of it. Most of the ATP, you're going to get the highest yield always in aerobic respiration. Now, with all of the breaking things down and building things up, and I'm like these catabolic and anabolic reactions, during metabolism, most of these reactions are what we call as amphibolic. It means they can go both ways. So you can take two molecules, my A and my B, and you can put them together in an anabolic reaction, and I can build them up and I make a larger molecule called AB. And there is a specific enzyme that helps do that. And then there's another enzyme that can take AB and reverse it and break it down into its separate parts. And I'm like, so it's breaking it down. And there'll be another separate enzyme that does that. So there's lots. You can have these same molecules. You can put them together, build them up, then you can go right back and break them back down. So this is kind of showing, that was my quick example. And I'm like, as you are constantly breaking things down, anytime you go in this direction on this diagram, you're breaking things down. And we can break down a lot of things. And I'm like, you can break things down in a catabolic reaction, but you can take any of these different products and put them together to build things right back up. And so it would be your anabolic reaction. So a lot of reactions go both ways. Not all of them, but a lot of reactions can go both ways. They just require their own specific enzyme. So we're going to end right there. A lot of information in that PowerPoint on all of the different types of reactions that are happening inside of the cell. And again, as always, if you have any questions, please feel free to send me an email and I will try to clarify anything you have.